Hello, and thanks for joining us today for the latest Technology Networks webinar, Securing Cell Therapy Success, the Role of PCR in Quality Control. I'm Sarah Whelan, a science writer here at Technology Networks, and I'm here to moderate today's event. I'm pleased to have Dr. Amanda Hayes and Dr. Xishan Zhu joining us today as your presenters. A warm welcome to you both. Amanda is a scientific officer and senior director at Bioagilitics Laboratories. She offers more than a decade of lab experience in multiple fields, including pharmacology, drug metabolism, immunoassays, immunogenicity, biomarkers, flow cytometry, as well as qPCR, and has supported programs through all stages of drug development. In her current role, she provides global scientific leadership and technical guidance at Bioagilitics. Xishan serves as Director of CMC Bioanalytical at Bioagilitics. Prior to that, she served in different roles in the BioCMC department at LabCorp in Greenfield, which has provided her with extensive experience and expertise on CMC analytical development and preclinical/clinical molecular bioanalytical development, as well as an in-depth understanding of regulatory compliance. So following the end of the webinar, we will have a Q&A session and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. And to ask a question, you should enter it into the box on the right-hand side of your screen and then click send. For any questions that we don't have time to get to, we will make sure you are contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. And please remember, you can ask questions at any time, even while watching the webinar on demand. So without further ado, I'll now hand over to Amanda and Xishan.
Thank you for that introduction. It's my pleasure to talk about cell therapies today with you. This is a quick disclaimer slide. And just a brief overview of our presentation today. We're going to be speaking about the role of molecular workflows, so quantitative PCR and digital PCR, and supporting one of the advanced therapeutic medicinal products, and that will be cell therapies. So we're going to talk more about the bioanalytical and CMC assays that are required to support these types of modalities. And today, briefly, I'm going to be talking about a type of novel modalities, as I mentioned. It's an advanced therapeutic medicinal product called cell therapies. Cell therapy products include cellular immunotherapies, cancer vaccines, and other types of autologous or allogeneic cells that can be used to treat certain therapeutic indications. They also include hematopoietic stem cells and adult and embryonic stem cells as well. And a branch of engineered cell therapies can also be used to repair, regenerate, and replace human tissue. But today we're going to be focusing on a branch of ex vivo gene therapies where cells are modified or manipulated to express specific genes or to alter the biological properties of those cells to then become a therapeutic. So that's what we'll be focusing on today. And the reason we're talking about this is because cell therapies are rapidly growing, with several cell therapies already approved by the FDA and the EMA. These therapies are projected to continue to grow, with many products in the pipeline, as shown here on this slide, in this graph. And you can see that CAR T cells are probably the most well-known of the cell therapy types that are either approved or being worked on, as well as several other cell therapy types. But you can see that several are in the pipeline, and many are continuing to work on these types of therapeutics because of their success in the clinic. So as I mentioned, there are already several FDA-approved cell therapies. These are mainly CAR T therapies, and several of them are known for the treatment of hematological malignancies or blood cancers. But what I wanted to mention on the slide is that even though that's a very niche indication right now, cell therapies are of increasing interest for treatment of other cancers and other diseases, including solid tumor cancers. And while there are several under development currently, pharma and biotech companies are trying to overcome some of the challenges that they face in this space, but is showing some good promise. So today, my colleague Shashin and I are going to talk about molecular applications that are used to support the development of cell therapies. And this is with respect to CMC and bioanalytical testing. And more specifically, we're going to cover the following assays. We're going to be talking about vector copy number assays, RCL and RCR testing, as well as adventitious agents for CMC support. We're also going to be talking about molecular assays used in the bioanalytical space for preclinical and clinical testing. And those are mainly assays, molecular assays to support persistence and biodistribution, as well as vector copy number in bioanalysis, and RCL and RCR testing and patient monitoring. And the two molecular technologies that we're going to be talking about today are quantitative PCR and digital PCR. And you might along the way see some parallels between the two. You can notice that in CMC and bioanalysis, some of these assays are used in both settings. So for a quick review of the technologies we'll be talking about, quantitative PCR has been around for many, many decades. Just real briefly, traditional quantitative real-time PCR is essentially the amplification of DNA. This DNA is extracted from a sample, PCR master mix, along with that extracted DNA, primers and probes, polymerase, and DNTPs are mixed together, and they are used to amplify the target sequence over multiple amplification cycles. And as the target sequence is amplified over these cycles, it generates a fluorescent signal that's captured during each PCR cycle. So as the number of copies increases during the reaction, so does the fluorescence, and it indicates the progress of the reaction as seen in this graph here. And for quantitative PCR, you have to have a standard curve with a known target concentration. And what this does is it allows you to back calculate the concentration of unknown samples based on the CQ or CT value that's obtained from that PCR reaction. So this technology has been used for a lot of different applications. And today we're going to talk about how it's used in cell therapies. Another platform that we're going to discuss is the digital PCR technology. It's a newer molecular technology, and it actually allows for absolute quantitation without the use of a standard curve. So in this workflow, this is an example of a digital PCR workflow. The sample is partitioned or it's split up into smaller reactions that are put into these microchambers, what you see here in this digitization picture right here. And it goes through a amplification process similar to traditional qPCR, where you have that sample in the microchamber that's amplified over multiple cycles, and that sample will either contain, or that microchamber is going to contain either 0 or 1 copy of that target, or multiple copies of that target. And it's determined, it also goes through as a fluorescent based readout, and the sample is either positive or negative based on the amount of fluorescence in that sample, and that correlates to a copy number above a certain threshold. And that's how the sample is quantified. So it doesn't require back calculating the signal to a reference standard. You just get your positive or negative droplets based on this threshold. And that tells you how many copies are in your sample. So to compare both technologies, as I mentioned previously, the main advantage of digital PCR is that absolute quantitation. It allows you to not use a standard curve to back calculate your unknown concentrations, versus traditional PCR uses that relative quantitation, and it needs that standard curve. The partitioning and the splitting of the sample in digital PCR reduces its susceptibility to matrix interference or PCR inhibition. And that is something that traditional PCR is susceptible to. Both platforms allow for multiplexing capabilities, and traditional quantitative PCR has a much wider dynamic range. So that's an advantage for samples that are expected to have high target copy numbers. However, digital PCR has greater precision at the low end, which is advantageous for rare event detection, allowing that detection with greater precision at the low end. So it's helpful for targets that are expected to have low copy numbers. Both platforms allow for use of the reverse transcription workflow, and the cost of instrumentation and cost of reagents differs slightly between the two, given that digital PCR is a little bit more new with respect to technology. And now I'm going to hand it over to my colleague Shishin, and she's going to talk about molecular assays for CMC support of cell therapies.
Thank you, Amanda. The application of PCR in CMC analytical support for cell therapy includes vector copy number determination. For genetically modified cell therapy product, for example, CAR T cells, transgene integration can potentially change the expression of the cellular genes and contribute to tumorigenicity. Therefore, transgene integration is an important safety parameter to measure. If the vector system directs transgene integration, the average number of the integrations per transduced cell, generally referred as a vector copy number or VCN, is required by FDA to be determined and reported on the certificate of analysis of each lot of the cell products. Quantitative PCR has been traditionally used for VCN assessment. The total vector copy number was determined by primer probe targeting the transgene. The total cell number was determined by cell counting, total DNA concentration, or the second set of primer probe targeting a single copy reference gene in the human genome. However, as Amanda just mentioned, that PCR quantification is based on a standard curve, so the results are directly impacted by the theoretical copy number determination of the standard material and also the preparation of the standard curve, which could cause high variation of VCN determination. Because of this disadvantage, some people started to try to avoid the use of standard curve, but use delta-delta CT method instead for VCN assessment. The graph on the left compares the approach of standard curve method and the delta-delta CT method. A delta-delta CT method using a duplex PCR assay, including two sets of primer probe, one for transgene and the other for a single copy reference gene, and no standard curve is needed. However, using this method requires a calibrated sample. This special calibrated sample is from a one vector copy cell clone, which is not easy to prepare, to purify, and to qualify. Compared to qPCR, digital PCR provides the advantage of absolute quantification and beta sensitivity and precision. Therefore, it becomes a preferred method for VCN assessment. The graph on the right shows the VCN determination results of a CAR T cell product using digital PCR. The result was tested over time and among different analysts. The great consistency of the results indicates a digital PCR is a robust tool for accurate assessment of average vector copy number. So the current standard approach for vector copy number assessment is through population analysis. In this approach, the genomic DNA is extracted from both cells, and the total number of vector copies as determined by qPCR or dPCR represents the average of the whole population. It doesn't give the true number of vector integration in each cell, and the VCN in each cell could be different. But the population VCN doesn't provide information of the cell-to-cell variability. This may have implications of product safety, and it may underestimate the presence of cell clones with higher value or a higher number of integrations. Those cells could persist and even replicate following infusion. It also lacks the resolution to monitor the change in the final product specifications. For example, the patient-specific donor cell material or the lot-to-lot variability of vectors may cause the change of VCN distribution, but the population VCN could stay the same. To overcome the disadvantage of population VCN, several groups started to measure viral vector integration at single-cell level. This slide showed a study design of VCN determination at single-cell level. So the human primary T cell was transduced with a lentivirus. The sample was loaded on a single-cell preparation system to capture individual cells in each microchamber on a chip, which then stand to identify the chambers containing the living cells. The captured live cell underwent the genomic DNA extraction, and then during the pre-amplification within the chip. The pre-amplified material was analyzed by duplex digital PCR. The spontaneous quantification of vector copy number and human reference copy number was used to generate a single-cell VCN database. The statistical analysis also provided the results of VCN distribution of the sample. As shown on the graph on the right, although the average VCN among all cells were comparable to the population VCN, there was a high level of heterogeneity in the VCN at the single cell level. The highest vector copy number per cell was like a 9. So it's very different compared to the average VCN number of 2. In addition, as the sample contains the mixture of transduced and non-transduced cells, the transduction efficiency value can be obtained from this single-cell digital PCR method. And this result shown in the graph E on the right lower panel. So this result is comparable to the result from the live imaging and the flow cytometry, suggesting this single-cell digital PCR-based method can provide another way to evaluate the transduction efficiency. Another application of PCR method in cell therapy is to test the replication-competent retrovirus or lentivirus. So gamma retrovirus and the lentivirus vectors are commonly used to introduce transgene into cell therapy products. Those retrovirus vectors are ideal for delivery and a stable integration of gene of interest into the host cell genome. However, they potentially pose risks to human health. One of the concerns is generation of a replication-competent retrovirus or lentivirus, RCR or RCL, which can infect non-target cells. FDA requires all cell-based products modified by this retrovirus vectors must be tested for RCR or RCL prior to treatment of the patient. There are different options for RCR, RCL testing. The most commonly used method are biological assay and molecular assay. Biological assay directly tests the infection and the replication of RCR and RCL. It is a cell-based assay, by incubating the cells with the testing material, which includes 3 weeks amplification phase and a 1 week indicator phase, with the following detection of virus using either ELISA to test the viral protein, P24, or molecular assay to test the function of reverse transcriptase. This cell culture-based assay is highly sensitive and has less chance for false positive results. However, it is time-consuming. It can take up to 4 to 6 weeks to get the results. Molecular assays, either qPCR or digital PCR-based, detects the gene sequence of the virus. These genes, for example, the envelope gene, VSV-G for lentivirus, or GALV envelope gene for gamma retrovirus, has to be integrated to the host cell genome during virus replication. For RCL testing in cell therapy product, the DNA will be extracted from the sample and then tested by PCR. Since PCR method targets the DNA sequence, if the sample contains any carryover DNA from the vector production cells or the residual vector plasmid, there's chance to get the false positive results. However, compared to cell culture-based assay, PCR method is much quicker. From DNA extraction to PCR, it takes only a couple of hours to get the results. It is also fairly sensitive. Therefore, the PCR-based assay is an alternative for rapid assessment of RCR and RCL in final products. The last application of PCR assay for CMC support for cell therapy is for adventitious agent detection. Adventitious agents including numerous microorganisms such as bacteria, fungi, virus, mycoplasma, and others. They are unintentionally introduced into biological medicine by, for example, the use of contaminated biological raw material for cell culture, the contaminated reagents for transfection or downstream purification, or the contaminated excipient during formulation. The contamination could also from environment during storage of raw material or during cell culture or media handling. As there's potential of these adventitious agents to cause serious harm to patients, manufacturers are required to strictly follow GMP guidance and regulations to ensure the risk is mitigated. Detection is critical for mitigation. Therefore, testing of adventitious agents is required at different stages of the manufacture process, from the starting material to the final end product. Just like RCR and RCL testing, there are multiple options for adventitious agent testing, including biological assays, in vivo or cell-based in vitro assay, and molecular assays. The advantage of biological assays is that they can screen for both known as well as unknown agents. However, these assays depend on the contaminating agents being able to amplify and have detectable effects in the testing system. Another major drawback is in vivo or in vitro-based detection is the compatibility of the testing with the biological drug itself. For example, if the biological drug has cytotoxicity, the cells in the testing system may die and the adventitious agents will not amplify, so the assay will not work. This problem sometimes can be solved by adding neutralizing antibody to the system. However, it makes the assay more complicated. In addition, as these methods can take weeks or even months. They're often the pacing item for the batch release. Molecular assays, PCR-based or next-generation sequencer-based, targeting DNA sequence, and the DNA will be extracted from the sample prior to testing. Therefore, these assays typically are not sensitive to the toxicity from the biologics to be tested. And also, these methods tend to consume a lot less of the precious product. So therefore, this method works together at the different stages of the production, provides a comprehensive strategy for advantageous agent detection. As a fast and highly sensitive and a specific assay, PCR is suitable for testing downstream in-process sample and the final product. Now I'm passing it back to Amanda.
Thank you, Shishin. So now I'm going to transition us into the molecular bioanalytical support of cell therapies. So you're going to see a lot of parallels from what Shishin just mentioned for CMC support in this section. So the first assay I'm going to be discussing is the characterization of biodistribution, persistence, and the cellular kinetics profile of cell therapies. So shown here is an example of a PK profile, and this is an example from the FDA-approved CAR-T cell therapy Kymriah, and it's shown here from a publication by Mueller and colleagues from 2017, and it's showing a cellular kinetics profile, and it's a representative from a patient with pediatric B-cell acute lymphocytic leukemia, showing the expansion phase of the CAR-T cells before day 10. And the PK profile was plotted here with data from qPCR assay that was measuring the transgene levels of the cell therapy as it goes through the unique phases of distribution, expansion, contraction, and persistence after that product was infused in the patient. And so to be able to collect this data, there are two different ways that you can measure cellular kinetics. One method is by quantitative PCR, shown here on the left, where the transgene DNA is quantified in the cell, and this is the inferred concentration measurement of the CAR-T. Alternatively, the CAR-T can be measured by flow cytometry, where an anti-CAR fluorescent labeled antibodies used to stain the CAR-T cells. And this is a direct measure of the CAR-T expression in circulation. Both methods can be used to understand the pharmacokinetics or cellular kinetics of CAR-T or cell therapies. Now, although both methods can be used, both have some advantages to whether a quantitative PCR assay is used or a flow cytometry assay is used. Both have very different workflows. They're very different technologies. Flow cytometry allows for the direct measure of the CAR expressing cells, as I mentioned. That platform allows for flexibility for developing high parameter panels that can provide more insight into the phenotype of the cell therapy. You can include markers for example, T-cell activation and exhaustion, and other markers of interest. For quantitative PCR, on the other hand, you can't get that level of information, but the major advantage is that the quantitative PCR assays are more sensitive, and they allow for the ability to detect very low copy numbers with greater precision to help understand, especially, and characterize the persistence of the cell therapy in circulation. So both can be used. And I'm going to focus on the molecular application here. So in order to set up a cellular kinetics or a persistence assay for a cell therapy, it's commonly performed by developing two assays or a duplex assay, one to detect the cell therapy target. And this is done to target the transgene, and the second assay, the transgene of the cell therapy. And the second assay is for a reference gene to normalize the reportable. The target assay can be against the backbone sequence of, for example, the lentiviral vector, or that target sequence can be designed to maybe a common construct for cell therapy transgenes, or it can be specific to that cell therapy transgene itself. And then the reference gene should be reliable and reproducible. You should be able to measure it. And the most important thing is when you choose your reference gene, you have to make sure that it's not susceptible to changes in expression in, for example, a disease state or other factors that could contribute to variability in expression levels of that reference gene. The matrices that are often collected for this type of analysis includes whole blood, PBMCs, maybe bone marrow aspirates, and even other matrices depending on the distribution profile of that cell therapy or even the route of administration of that cell therapy. So my colleague Shashin already went through the basics of RCL and RCR testing in the CMC environment, but what I wanted to mention here is that patient monitoring for RCL and RCR is also performed in the bioanalytical setting, and especially where lentiviral vectors are, for example, for RCL testing, where these vectors are used to introduce transgenes into the cell therapy. So it can pose a safety risk in patients by possibly infecting non-target cells, and thus it's a requirement to test for RCL to monitor these patients throughout administration of that cell therapy. And assays are typically set up similarly in the CMC space, where you're detecting the envelope gene, for example, a VSVG sequence or similar for RCL testing. So very similar. Some of the challenges of understanding cellular kinetics and performing some of these assays is integration of data from multiple platforms or measurements is sometimes needed. One of the biggest hurdles, I think, in this space, especially in the molecular space, is there is an absence of bioanalytical method validation guidance from regulatory agencies or health authorities for PCR, which adds some ambiguity to how these assays are developed, how they're validated, and what acceptance criteria are appropriate for them. Another challenge is prior to cell therapy infusion, patients are typically lymphodepleted, which can cause hurdles with sample analysis with low cell number counts, and it causes some issues and some challenges with data reporting. I mentioned that flow cytometry and PCR can both be used for cell therapy pharmacokinetics or cellular kinetics characterization. It's also challenging and difficult when you're measuring these by these very different platforms and trying to understand the concordance of results and the differences in reportables. And finally, another challenge with PK assessments in this space is assessments like incurred sample reanalysis. That's a historical PK assessment. Sometimes it's difficult and challenging to reproduce in a molecular setting and in a flow cytometry setting for that matter. So it causes a lot of confusion in the industry as well. So I'm briefly going to talk about the molecular workflow, regardless of the purpose, whether that's CMC or bioanalytical. When you have a molecular assay, the extraction of nucleic acid from the drug product or the patient sample is part of the method, and it can be performed with a variety of techniques, including isolation and purification with silica membranes or magnetic beads. These can be manual processes or automated, which are often nice to have with high throughput sample extractions. So after your nucleic acid's extracted, you have to determine the concentration, and this is usually measured and gives you a mass per volume unit to work with your nucleic acid. It can be measured with a variety of different platforms and different technologies, including absorbance or fluorescence methods. And you can also get additional parameters on your nucleic acid, like understanding the purity or your integrity as it's used in the downstream PCR application. So as I mentioned, there's no validation guidance that is for PCR that is available. So the validations are fit for purpose, and they are similar to any other bioanalytical or analytical method validation, where the performance of the assay is characterized first by understanding its reproducibility, linearity, specificity, matrix interference, robustness, and stabilities for your method. So this is common across most methods in regulatory analysis. And the assay purpose is really what should drive how to set up the experiments for understanding these performance characteristics, and more importantly, what acceptance criteria are appropriate for your method. So all of these are essentially applicable for your molecular workflow. And today I'm going to very briefly go over just a few of these performance characteristics in the next few slides. I could probably spend another hour speaking about these different performance characteristics for molecular assay validations, but I'm just going to touch on a few today. The first is precision and accuracy. Reference material is used for assessing precision and accuracy of your method, and this is ideally the drug product or the cells in this case. If those are not available, which oftentimes they are not during method development and validation, a surrogate material can be used like plasmids or even synthetic DNA fragments like G blocks. And how to characterize precision and accuracy is done by using multiple QC levels throughout the dynamic range of the assay. And this is done with several determinations, ideally 3 over multiple runs and multiple days. And acceptance criteria should be used based on the context of use of the assay, as well as how it performed in method development. An ideal assay should have precision of your QCs around 30% CV and 50% at the limits of quantitation, an accuracy of -50 to 100% for quantitative PCR, or 30 to 50 for percent RE for digital PCR. And I do want to stress that these are suggestions, and again, acceptance criteria should be suitable based on the context of use of your assay and how it performs in method development. And another thing I want to stress with these suggestions or these recommendations listed on the slide is it does differ, and they seem a little bit wider if you're used to, for example, a platform like ligand binding assays or LCMS, where those acceptance criteria are known to be much tighter or smaller. And the reason for this is when you think about molecular workflows, is there's quite a bit of variability that can come from the extraction process, the PCR process, different analysts. That extraction especially is a large piece of the workflow that can introduce some variability. So it's important to understand that these assays do perform quite differently than some of the other platforms that we're commonly accustomed to. So another performance characteristic that I'm going to touch on is PCR efficiency and linearity. It's commonly assessed in molecular assays, similar to other workflows and platforms. And typically this is assessed with the calibration curve in quantitative PCR, or in digital PCR where you don't have your calibration curve. You can use a dilution series that spans your dynamic range. And this is assessed over multiple days and multiple runs. And the recommended acceptance criteria is an R-square value that you get from your linear range of the assay that's greater than or equal to 0.98. This corresponds to a slope of your curve between -3.1 and -3.6, or a 90 to 110% efficiency. And this is all determined with an equation that's commonly used to assess linearity. And again, I will mention these are recommendations. The acceptance criteria should, although, be driven by method development and how your assay is behaving, as well as the context of use and how you're going to use it for testing. Lastly, the final performance characteristic that I'm going to touch on is sensitivity for molecular assays. It can be assessed by defining 3 different pieces of your assay: the limit of blank, the limit of detection, or LOD, and the lower limit of quantitation, or LLOQ. And these can be determined by a variety of ways. It can be determined by mathematical equations or by different statistical means. And understanding the difference between these 3 different limits really helps us to understand the background of the assay and specificity. It helps you understand the analyte that can be detected, although not quantifiable in this case, if it's below the LLOQ or above the LOD. And finally, when you determine your LLOQ, that is the lowest value of the analyte that is reliably quantified with accuracy and precision from your assay. So understanding the interplay of these 3 values can help with understanding the level of sensitivity. And there's a lot of discussion in the industry as to how to appropriately measure these, and although there's no right or wrong way, it's important to understand how they play together. And with that, I'm going to conclude with just hoping that you understand or take away that molecular assays are important for supporting bioanalytical assays and also for CMC characterization for cell therapies and drug development. My colleague Shishin and I talked a lot about the different assays that can be used in this space. And hopefully you also were able to see a lot of parallels between the bioanalysis and the analytical CMC world. And so with that, bioanalytical scientists and CMC scientists can gain insights from each other while they develop these and validate these molecular assays that are used to support their respective needs. And with that, we would be happy to take any questions, and thank you.
Thank you very much, Amanda and Shishan. I'm sure our audience will agree that there are some excellent points raised during the presentation. So we will now move on to the Q&A session. So to submit a question to our speakers, you should enter your question into the box on the right-hand side of your screen and then click send. We've had several questions submitted already, so I will move straight on to these now. So Amanda and Shishan, if either of you would like to jump in on some of these questions. The first one is, can you use the same VCN assay for CMC and clinical sample analysis?
That's a great question. So, they're essentially the same assay. You could use possibly the same primers and probe sets if you're setting up the assay. I think the main difference is both assays have to be validated for their context of use. So in the CMC setting, it's different than when you're validating for a clinical sample analysis. And the workflows are going to be slightly different because when you think about extraction, you're going to be extracting DNA from, say, a drug product in the CMC space versus extracting DNA or nucleic acid from the patient sample. So you're going to have some background matrix in that sample, whether that's whole blood or PBMCs or other. So there are some parallels that can be used. However, the assay validations are very specific for their own application. And Shishin, I don't know if you have anything to add with that respect.
No, I think you covered it well. And yeah, the sample matrix will be different. And also, I think for the validation parameters, for the CMC VCN testing, we usually have the pure, like the drug product sample. So the sensitivity is not a great concern for the CMC assay, but for bioanalysis, for some samples, it may be a concern. It's a focus during the method development.
Thank you both. So the next question is, when would you need to use the culture-based or PCR-based assay for CMC support?
Okay, I can take this. So since cell culture-based assay is highly sensitive and has less chance for false positive results. So it is a preferred method for like ICR, RCL testing. For GMP manufacturing, FDA requires RCR, RCL testing has to be performed using cell culture-based assay for the end of vector production cells and the vector supernatant, and also for the final product if times allow. However, for many cellular therapies, which requires the infusion of fresh cells, it doesn't give you like enough time to complete a full cell culture-based RCR, RCL testing prior to infusion. So for this reason, a PCR-based assay is an ideal acceptable method for a rapid assessment prior to a fresh product release. Sometimes we call it conditional release testing.
Thank you. The next question is, do you need the same level of sensitivity for a VCN assay for CMC and bioanalytical?
Yeah, maybe I'll take a stab at this. So she should mention that in the last the first question, I believe. So the level of sensitivity is very dependent on the context of use of that assay, and it could be different in a CMC setting versus a bioanalytical. And I'll speak on behalf of the bioanalytical testing as Shishin mentioned, when you're trying to characterize the persistence part of the cellular kinetics profile in patients, you really need a very sensitive assay because you're trying to detect the lowest copy number that you can of the cell therapy in circulation at that point. So sensitivity then becomes a very critical parameter to test and validation. And that might be a little bit different in the CMC world.
Fantastic. And I'll move on to the next question. So this one is, can you use the same RCR or RCL assay for CMC and clinical sample analysis?
Yeah, that's a great question, and very similar to the answer that I gave for the first question is, can you use the same vector copy number? So in this case, the same RCR or RCL assay. And the answer is pretty much the same as it's going to depend on how you use the assay is based on how you're going to validate it. So it's going to be validated for its context of use, which it would be different in maybe your acceptance criteria might be a little bit different in the CMC setting versus the clinical sample analysis. And more importantly, you're extracting DNA from a nucleic acid from human matrix for clinical sample analysis versus extracting nucleic acid from the actual cells or the drug product in the CMC setting. So some differences there.
Thank you. And the next question also touches on this acceptance criteria. So this one is, what kind of acceptance criteria would you use for precision and accuracy for VCN or RCR, RCL assays?
That's a good question as well. I think it depends on your how your method is performing and during method development and as you're optimizing the assay, and really understanding how your precision and accuracy looks in method development should drive what acceptance criteria you're going to use in your validation. And I did, throughout the presentation, I gave some kind of recommendations for what an acceptable criteria could look like for molecular assays. However, at the end of the day, you just have to make sure that you're validating your method, and more importantly, that the acceptance criteria that you're using for your method is going to support how you're going to use that data. So whether that needs to be more stringent or a little bit more loose, it really depends on how that data is going to be used.
Great. And is there a preference for using dPCR or qPCR for VCN or RCL testing?
I think, yeah, as Amanda mentioned, and I also included in the presentation, so there's difference between digital PCR and the qPCR. So digital PCR doesn't require the standard curve. So you don't have to maintain a reference standard material, and during the life cycle of the method, you don't have to bridge different lots of the reference standard material. So that's definitely an advantage. But other than that, I think for the sensitivity, precision, they're comparable. And so if the instrument is suitable for your purpose, you can use the instrument you already have. But I think for some if you're doing a method for a long time and you don't want the burden to manage the reference standard, the digital PCR definitely have some advantage on that perspective. Amanda, do you have anything to add?
Yeah, I think that's very similar in the bioanalytical world as well. You can have a qPCR or a digital PCR assay for vector copy number or RCL, but I think digital PCR really offers that higher level of precision at the low end, which becomes a nice advantage when you're looking at vector copy number or copies of your CAR T or your cell therapy in circulation in that persistence phase where you're trying to detect, the lowest amount you can. So digital PCR offers that advantage there.
Thank you. And the last question I have here is, what happens if you have positive RCL samples from your drug product?
I really hope you don't get any positive RCL results for your samples. But if you do get it, so first that fill your product release specification, so it will be out of specification investigation. You need to confirm this positive result is valid. And if it is valid, then you need to do the further investigation. You have to probably run cell culture-based assay to confirm the result is not is a real positive, not a false positive result of your sample. And if really is a positive result, then the additional investigation has to be done to find out your manufacturing process. So that will be a very significant issue to deal with. But with the technology improvement, I think with the third generation of the CAR T cell manufacturing process, the chance to get the positive RCR results is very low.
Fantastic. Well, thank you very much to you both. That is all we have time for today. So just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. And you can continue to ask questions even while watching the webinar on demand. A certificate of attendance is now available to download from the Handouts tab on the right-hand side of the webinar platform, and the webinar will now be made available on demand. So if you have any friends or colleagues that you think may be interested in the webinar, please feel free to share the link with them. And thank you again for your time.